It's now my pleasure to introduce our next speaker, Dr. Paul Henderson. Dr. Henderson is an assistant adjunct professor of hematology and oncology at the Department of Internal Medicine at UC Davis Health System. A toxicologist, his research is focused on new technologies to determine which patients will benefit from cancer drugs, a project that promises to improve outcomes from many patients for whom chemotherapy has previously been considered too risky. Dr. Henderson took first place this year in the UC Davis Big Bang Business Plan competition, winning $10,000 to advance his research. He recently founded a company called Accelerated Medical Diagnostics to market the test that he and his colleagues have developed, and he secured the commercial funding to move that forward. Dr. Henderson leads a multidisciplinary team that includes faculty and students from UC Davis's School of Medicine's Cancer Center, the School of Veterinary Medicine, and the Graduate School of Management, as well as the Lawrence Livermore National Laboratory, the National Science Foundation, and several private companies. Please join me in welcoming Dr. Paul Henderson of the UC Davis Health System.
Thank you, Professor Montañez, for that very kind introduction. I took the teleprompter option today because I've never used one before, and I can tell already it's awesome. And I used a TRS-80, by the way. It's an enormous honor for me to be here with all of you today to help launch the academic year at UC Davis. Thank you, Dr. Katehi, for your leadership in helping UC Davis to become a hub for innovation and for creating the kind of environment here that inspires scientists like me to tackle the big problems in science and medicine that can improve health for all. As you know from Professor Montañez's introduction, the goal of my work here at UC Davis is to solve a vexing problem for cancer specialists and their patients. Although we know that chemotherapy can kill cancer cells and effectively cure many patients, we don't know which patients will ultimately benefit. In some cases, the toxic drugs actually harm or even kill the patients before they even get the chance to kill the tumors that they're trying to target. My goal is to develop a test that can determine whether chemotherapy will actually work to kill an individual patient's cancer before that drug is administered in the usual dose needed to be effective. This is called personalized medicine. It's a really hot topic right now. This concept, though, for our particular application, was born 10 years ago as the result of the kind of collaboration that our leadership hopes will define and distinguish our institution for generations to come. In this case, it was Cancer Center Director, urologist Dr. Rafe DeVere-White, working with Biosciences Division Leader Ken Turteltaub at Lawrence Livermore National Laboratory. Their idea was to use something called accelerator mass spectrometry to give patients non-toxic doses of chemotherapy without exposing them to dangerous radiation and to predict whether or not they would respond to the therapeutic dose of the drug. Now, I'm going to hold the teleprompter here for a second because that was a big sentence. Basically, Livermore has technologies that say if a terrorist ever handled plutonium in their entire life, a tiny bit of that plutonium gets sequestered into the bone, and it slowly leaches out. They can tell if using this technology whether someone has handled plutonium once in their life. There are also technologies available where if someone, say a drug dealer, handled cocaine, they didn't even use the drug, they could detect it in their hair. These are the kinds of technologies that are developed against terrorism and the war on drugs, and it's about time to bring these types of technologies to bear on medicine, where they can not only be used to sniff out a rare few really bad people that have high consequence intentions, but that can help the general population, such as the 40% of you who may someday be diagnosed with cancer and the 20% of the people in this audience, unfortunately, that will actually probably succumb to the disease. I came to this project as a toxicologist with an interest in the damage that certain chemicals can cause to DNA. And those are the same kinds of damage, it turns out, that causes mutations in cells and causes cancer, but also cancer drugs also bind to DNA and damage the tumor DNA. It's DNA's ability to repair itself that has strong relevance to cancer, and the ability of cancer cells to resist certain treatments such as radiation or chemotherapy, comes directly from that characteristic of all of our cells. In a patient whose cells resist treatment, these therapies cause far more harm than good. In my first work as a researcher at the National Laboratory System and now at UC Davis Cancer Center, we are using an accelerator mass spectrometer to test the effect of tiny, tiny doses of a common cancer drug called carboplatin on the cancer cell's DNA. And this is in patients. It's not just in petri dishes with cells. These DNA-damaged products are called adducts. They can be measured in a tumor in order to predict the response to chemotherapy. We so far tested this approach in 6 bladder and lung cancer patients. I now co-lead the research project with Dr. Chong Shan-pan, a physician and cancer researcher at the UC Davis Cancer Center. Since then, the partnership between UC Davis, the Cancer Center, the National Laboratory System has also grown to include a wide variety of interests, including the National Institutes of Health, which partially funds this work, the UC Davis School of Veterinary Medicine, where this experiment is being replicated in dogs with canine bladder cancer patients. Dogs with bladder cancer, they look and respond much like humans with bladder cancer patients, so it's a great model. The National Science Foundation's Partnership for Innovation, itself a multidisciplinary collaboration, has actually funded our company to participate in this work and also provided students and mentoring. The UC Davis Graduate School of Management, which helped to develop the business plan for our company. Vitalia Sciences, a local Davis company that actually owns an accelerator mass spectrometer and it uses it for pharmaceutical research, is also a partner in this work. Response Genetics, a company whose technologies are also helping to personalize cancer treatment, as well as the Sacramento Angels, a group of Sacramento area venture capitalists. Well, they're actually angels. They hate being called venture capitalists. But they provided us with sage advice, and they've even tried to fund us, and maybe someday they'll wise up and actually cut us a check. In 2008, Dr. Pan and I launched our company, Accelerated Medical Diagnostics. We're now poised to commercialize this test for use in cancer patients. All told, we have more than a dozen people working on solving this problem, and it could not happen outside an environment in which collaboration is rewarded and encouraged, such as it is here now at UC Davis. Ours is an example of the importance of aggressive networking and uniting a diverse group of experts and resources to develop the solutions to big problems. Of course, as my mentors here know, having an entrepreneurial spirit and developing multidisciplinary partnerships are just the beginning. We have many big hurdles yet to clear, including the need to raise funds for clinical trials and to win support from the Food and Drug Administration as well as other regulatory agencies. I am optimistic that we will eventually succeed in breaking the barriers that have prevented so many cancer patients from benefiting from these life-saving treatments. I'm lucky to be part of UC Davis, where the big ideas are nurtured and the innovation is now available and developing here to enable, through strong leadership, a bold vision and a commitment to translate our discoveries into exciting new tools to develop to improve the lives of our patients. Really, thank you very much for this opportunity to share our vision with you.